Hi, can the folks at the back hear me okay? Perfect. Okay, sorry, that's just an awful joke, but I've been speaking to my laptop for about an hour and a half now, so I hope you guys will understand. My name is Harsha. I'm really excited to be here today, and in today's talk, I'm going to be taking you through this really neat little story that's been unfolding in our lab over the last year and a bit. We've been looking at some populations that we maintain in allopatry in the lab, and we're asking questions of whether or not they've undergone reproductive isolation. So the story actually begins with questions of aging and senescence. Back in the late 80s and the early 90s, this lab in UC Irvine, run by Dr. Michael Rose, was interested in figuring out if selecting for extremely late stage reproduction or extremely accelerated reproduction produced correlated responses in aging and senescence. They took this population known as the IVs, split it into five replicates, which they named baselines B1 through to B5, and subjected them to two selection regimes. In one selection regime, they were selected to reproduce extremely late, and in the other one, they were selected to reproduce very early. These populations have since gone on to be called the Cs and the As, and are now maintained on much more standardized regimes rather than active selection program. The Cs are cultured once in 28 days in non-overlapping generations, and the As are cultured once in 9 days, also in non-overlapping generations. These populations produced a wonderful tool to study aging and senescence, and then went on to become really useful in studying correlated responses and trade-offs. They have since also been used to study artifacts in selection regimes and accidental responses in phenotype. Most recently, PhD candidate in the lab, Joshua Alpern, has been using these populations as an excellent tool to figure out the genomic differences in developmental pathways that the A's and C's undergo. For this research project, we were interested in figuring out if there was any levels of reproductive isolation that were evolving between the C and A selection regimes. We know from their generation time roughly how many generations these populations have been diverging for. Uh, Given this knowledge, we can roughly claim that they've been separated for about 15-20 generations, which is a microscopic period of time for macroevolutionary trends such as speciation to take place. Uh, so it was of deep interest to us to figure out if there was any barrier to gene flow that had evolved between these populations, even in this short period of time. For replication and additional statistical solidity, we repeated all the assays that were conducted on the ones with the threes and the fives, the C3 and A3 populations and C5 and A5 populations. To study reproductive isolation between the A's and the C's, we considered all the possible stages at which barriers to gene flow could exist between them. We were not very concerned with temporal isolation given that we were working with Drosophila species, but what we did know is that these populations undergo complete habitat isolation because they're maintained in allopatry in the lab. So moving forward, our strategy to study the levels of reproductive isolation was to create artificial hybrid zones where we created very controlled interactions between the A and C populations and looked for barriers to gene flow. To begin with, we started with mate choice experiments and mating observations to see if there was any level of behavioral isolation that prevented mating attempts or mechanical isolation that reduced the chance of mating from being successful. Having looked at that, we looked at the hatchability of eggs and the viability of the hybrid spawn of these eggs. The point of looking at hatchability was to look for post-mating pre-zygotic barriers where incompatible gametes or reproductive tract seminal fluid interactions prevented fertilization from happening. Having looked at viability, we then looked at reproductive fitness to address further post-zygotic barriers. We studied F1 hybrids and looked for reduced hybrid fertility, and also looked at F2 offspring to see if there was hybrid breakdown in subsequent generations. We began with assays to determine if the A's or C's showed a significant preference in mate choice. To ensure that these assays were controlled for mating status, we conducted all our assays using virgin flies that were sexed and separated shortly after eclosion. In order to ensure that low resource availability did not compromise any of the behavioral decisions of these flies, we also reared them all in high food resources. Focal flies in both the male mate choice tests and the female mate choice tests were provided with two flies of the opposite sex, one from each selection regime, from within the same replicated block. So, for instance, if we were testing the mate choices of A1 females, they were provided with a single A1 male and a single C1 male, and the mated male's population identity was recorded, along with copulation duration and the latency to mating. The male mate choice assay and the female mate choice assay were nearly completely identical, with the exception of the method we used to identify the mated individual's population identity. Females of the A and C populations are so distinct in size that it was easy for us to distinguish the mated individual's population identity 100% of the time. The same was not true of the males. In order to mark the males, we fed them small amounts of food dye which colored their abdomen blue and red in this experiment, which allowed us to determine which population the mated male came from. So before I get to the results from that experiment, I wanted to discuss some potential scenarios that could arise from such a mate choice experiment. Now, it's obviously possible that both the individuals of the A population and the C population mated completely randomly, and the number of homotypic matings and heterotypic matings were alike in both, which would be a clear sign of there not being any reproductive isolation between them. However, even within scenarios of assortative mating, it's not necessary that we are always looking at reproductive isolation or speciation. To begin with, let's consider the scenario on the right where all the flies involved display a preference for a specific body size, say large individuals. In this case, the Cs would mate homotypically, always picking the larger individual, the C fly, and the As would mate heterotypically, also always picking the large individual, which is the C fly. Such a result has actually been displayed in the past. Shampa Ghosh and Amitabh Joshi in 2012 worked on a system very similar to our own, with two fly populations that were selected for late stage reproduction and early stage reproduction, and showed that both the females of both the populations essentially preferred large males in order for mating. The other kind of scenario of assortative mating, which would actually suggest levels of reproductive isolation and barriers to gene flow, is when both populations choose to mate homotypically rather than heterotypically. Such a situation is true of melanogaster and similants within the Drosophila family itself. So, do any of these models or scenarios actually apply to the data that we obtained from our mate choice experiments? We began by analyzing our data across replicates for heterogeneity, and having found none, we display our results by selection regime, pooling the data over replicates. In the female populations, both the A's and the C's display deviations from random mating and a preference for conspecific mates. In the male populations, only the A males display such a preference for conspecific mates and a deviation from random mating, whereas the C males display almost completely random mating behavior. We interpret these results as the existence of barriers to gene flow at the premating level between these two populations, driven by sexual selection and sexual conflict. The female data is rather easy to interpret, with there being clear homotypic preference in both of these populations. The male data is not quite as straightforward, especially the C males' decisions to mate at random rather than mating preferentially with C females that are larger and by default more fecund is something that requires explanation. There are a number of reasons why this could possibly be happening. The first and foremost of these is that when a virgin male is presented with two virgin females, given a strategy of mating as multiply as possible, the male may not exert any choice at all. The second possibility is that is rooted in the different evolutionary histories of the A and the C population. Given the greater levels of sexual conflict in the larger C populations, it is likely that C females have evolved much greater levels of mate harm resistance and the capacity to resist courtship attempts by males. In this scenario, the C male could be preferring to mate with the C female, but might settle for whichever female is easiest to mate with in the given sample. This might also very well be the reason why the A males seem to display an extremely large preference for A females over the larger and more fecund C females. It could simply be that the C females are so capable of resisting mating attempts by the A males that are not evolutionarily equipped to deal with such large females that the A males end up mating with A females by and large. Having looked at pre-mating isolation, we then went on to produce hybrids to study hybrid viability and hybrid reproductive fitness. Now, I wanted to show how these crosses actually work to produce all the populations simultaneously in the same generation. But since I don't have that kind of time, I'm just going to display all the populations that were actually involved in this experiment. We had C parental flies and A parental flies on the left and rightmost corners. F1 hybrids produced through reciprocal crosses between the C parentals and the A parentals, and F2 hybrids that were produced by selfing the F1 hybrids. Flies from each one of these treatments were then observed for measurements of viability in each stage of their growth. Egg to larval hatchability was observed. Larvae that successfully pupated were counted. Pupae that successfully closed were then counted. and we produced individual measures of stage-wise viability and a net viability measure. Our hatchability and viability data from this experiment allow us to confirm that there is no reduced hybrid viability at any of the stages measured. Two key takeaways exist in this experiment. The first, hatchability across all treatments hovers between 95 and 100%, and this confirms for us that there is no post-mating pre-zygotic reproductive isolation between these two populations. The second key takeaway is that even though there are significant differences in net egg to adult viability, it is such that the hybrid treatments, 3 out of 4 hybrid treatments, actually have significantly higher viabilities than the parental treatments. This confirms for us that at the stage of viability, there is no reduction in hybrid fitness. Having confirmed no reductions in hybrid viability, we went on to study reproductive fitness. In this experiment, as with the mate choice experiments, virgin flies reared in high food conditions were used. Single focal flies were combined with 4 competitor flies and 4 target mates. Competitors and target mates were homozygous for recessive mutation BW, which confers a brown-eyed phenotypic marker, which allows us to record the proportion of offspring that belong to the red-eyed focal fly. In order to use an outgroup that is common to the C's, the A's, and all potential hybrids that ensue from them, we use the IV baseline ancestor and backcross the BW mutant into those lines, which were then called the IV browns. We allowed the focal fly, the target mates, and the competitors to mate for a period of 2 days together, after which they were transferred into a fresh vial to lay eggs for a limited period of time. The offspring that were produced from these eggs uh, were then carefully observed under a microscope, and the proportion of red-eyed offspring was recorded. What kind of fitness patterns might we expect from these populations? They may follow Haldane's rule, which postulates that the heterogametic sex tends to suffer a greater depression in fitness between populations undergoing post-zygotic reproductive isolation. In fact, this is especially relevant to Drosophila, where the Haldane's rule is so ubiquitous amongst divergent populations that it is treated as a common diagnostic step in the progress to post-zygotic isolation. They may display hybrid breakdown, where parentals are crossed together to produce healthy and fertile F1 hybrids, but F1 hybrids self together produce extremely low fitness F2 hybrids. This is the case with the cotton strains Gossypium hirsutum and Gossypium barbadens, which are capable of producing highly fit F1 hybrids but extremely low fitness F2 hybrids. We might also see a pattern where there is no decline in fitness whatsoever between the parentals, the F1 hybrids, and the F2 hybrids, and this can happen in spite of high levels of premating reproductive isolation, as seen in the Darwin finches of the Galapagos Islands. So which of these patterns apply themselves to our data, if any? To orient ourselves to this graph, we need to know that orange box plots depict female fitness data and blue box plots depict male fitness data. There's an immediate temptation to think that Haldane's rule is at play because at every population, the male fitness is depreciated relative to the female fitness. This is not the case, and let's put a pin on this temporarily and get back to it. Let's begin by looking at the parental fitnesses. A males have lower fitness than C males, and A females have lower fitnesses than C females. This is understandable and even expected given the evolved body size differences between these populations. Hybrid fitnesses of all the hybrids fall roughly between the A fitness and the C fitness of the respective sex. Within hybrids themselves, there is a trend for F1 fitness to be higher than F2 fitness, but when subjected to statistical scrutiny, only 2 out of 12 comparisons across all replicates actually suggested a significant difference between F1 and F2 fitness. Therefore, there might be a trend of F2 fitnesses being depreciated relevant to F1 fitnesses, but this is not significant and we don't have any confirmed evidence of hybrid breakdown. So we've confirmed that hybrids have roughly intermediate fitness to the parental A's and C's, and that hybrid breakdown, while a tentative trend, is not a confirmed phenomenon. We need to circle back to the elephant in the room, which is the reduced male fitnesses. The reason we don't believe that this is a case of Haldane's rule is because the depreciation in male fitness is across the board and not restricted to the hybrids. Why we believe this is happening has to do with the usage of the Ivy brown populations as competitors and target mates. The Ivy browns are a common ancestor to both the C's and the A's and have diverged from them in allopatry for an even longer period of time. This does not reflect on the female fitness data, as Ivy brown males probably do not distinguish between Ivy brown females and the A, C, or hybrid females when trying to find a mate. On the other hand, Ivy brown females might be much more choosy in exercising mate choice and might discriminate against A, C, or hybrid males and instead might mate with the Ivy brown conspecific males in the competition wilds. As a consequence of this, we believe that male fitness across all the treatments has been depreciated, which has less to do with post-zygotic uh, reproductive isolation between the Cs and As and has more to do with the pre-zygotic reproductive isolation between the As, Cs, and the Ivs. So overall, through this experiment, we established the existence of bidirectional premating reproductive isolation barriers. We managed to show that there are no post-mating pre-zygotic barriers, and in terms of post-zygotic barriers, while there is no reduced hybrid viability or reduced reproductive fitness in the females, the male data is inconclusive because even though the pattern seems to be reasonably consistent, the male data is significantly depressed from the expected levels of 1/5 of the proportion of offspring. In terms of hybrid breakdown, we're looking at some tentative trends that suggest that F2 fitnesses are lower than F1 fitnesses, but here as well, we don't have significant significant relationships that allow us to confirm a barrier to gene flow. So having significantly overshot my time here, I'd like to make a super quick shout out to Chloe Robinson and Dean Haydon, two undergraduate thesis students who have worked extremely hard on this project, both in white here. I'd like to thank the entire lab group and my supervisor, Adam Chippindale, my funding sources, NSERC, and my university, and BioRender for most of the amazing graphics that you've been seeing on this presentation. Given that we don't have a chance to properly network, this is me, and I'm happy to answer any questions that you'd like to shoot out to me, and I'm happy to talk about potential collaborations that anybody might be interested in.